Good afternoon, everybody. Everybody can listen to me well? Is that better? Yeah. Thanks again, Ursula. That was wonderful. And thanks, a Woman in Science Salk program, for inviting me. Thank you all for coming. It's such an excitement to be able to share my research with you. Let me get my water. Today, I'll tell you a bit about one of the aspects of the research we do in the lab of Fred Gage at the Salk Institute. And specifically, I'm going to tell you how do we research human neurological diseases using a new type of model that I will explain you how. Particularly, I'm going to talk to you about autism spectrum disorders. And autism is a neurodevelopmental condition in which the kids to be part of the umbrella of autism spectrum disorders, a child will show a combination of 3 different clinical characteristics. One of them is impaired social interaction. The other one is impaired language acquisition and communication, and also repetitive and restricted behaviors. And it's a few facts for us to try to understand this disease a little bit better. Over 1% of children in the US have autism spectrum, are under the umbrella of autism spectrum. Approximately 1.5 million of Americans. The cost per child affected with autism over a lifetime is 3 to 5 million estimated. Autism costs America approximately 60 billion annually. And the Autism Society of America has estimated that has proposed that if we could reduce this cost by 2/3 if we invested more in awareness, in diagnosis, and in therapies. We don't really know what causes autism, but what we know is there is a very big genetic component. There is also an environment component, but we don't know how those two interact with each other. And if you take twins that are identical, and we compare one twin that have autism, the chances of the other twin to have autism is up to 80%. So that indicates to us that there is a big genetic component, meaning it's on your DNA. And lastly, there is no cure and no therapy for autism. There are only behavioral interventions that alleviate the symptoms a little bit, but there is definitely no cure and no therapy to date. So how to study autism? Of course, we cannot really pick the cells into the child's brain. Unlike many other cells in the body, you can study, for instance, blood cells or skin cells, but with autism, you can't really take any cells from the patients without causing major effects or major problems. So traditionally, the way neurological diseases were studied is we would get patients' brain samples after the patient passed away. And we have got a lot of interesting insights from this strategy. But two major complications, 2 major problems with this strategy, or limitations of this strategy, is that we cannot get functional information. When we get post-mortem samples from a brain after the patient has passed away, you cannot get any functional information. You don't know what the cell was doing because you are pretty much looking at a dead tissue. The second major limitation is you're getting a sample from a patient that has passed away, So you're looking at endpoint of the disease. You don't know really what causes the disease from the beginning. And remember, autism is a neurodevelopmental disease, so it happens during the development, sometime where the kid is developing, most likely in the uterus. The other way to study autism is to look at some peripheral tissue, for instance, blood. So you can get some insights from the blood, but the blood is not really the target tissue where the disorder manifests. And lastly, we can have research animals give us some insights on the genetics of autism, but research animals can't really tell us a lot about typical human behavior, such as language acquisition or social interactions. Mice can't really talk. On top of that, mice, there are structures in the human brain, and a lot of them have been implicated with autism spectrum disorders that are not present in the mouse brain. So you can't really compare. So our idea, or what we propose, is to use stem cells as a new way to research autism spectrum disorders and other neurological diseases for that matter. And what are stem cells? Stem cells are very, very special type of cells that have two main characteristics. Stem cell is able to multiply and make daughters that are exactly the same as stem cells. So we call that self-renewal ability. And also stem cells have the ability to make cells potentially from all the cells in our body. And we call that the ability to differentiate. And that's how they look like under the microscope. They're very tiny and they grow together in colonies. Now, we can use these stem cells to research autism spectrum disorders. And the way researchers, scientists research subjects is we use models. So we use model. We're going to use stem cells as model to study autism spectrum disorder. And what is a model? A model can be a fashion model. Gisele Bichan, Brazilian, I know her. A model can be a little train that kid will use to play. A model can be an architect's model. And what all these models have in common is that they are simplified representations of the reality. And that's exactly what we are proposing to do with stem cells. We want to use stem cells as a simplified model of autism. So what kind of stem cells we're going to be using? We will use a special type of stem cell called induced pluripotent stem cells. And the reason why they're called induced is because they were not stem cells to begin with. They were cells from the body, the ones cells that are easily accessible as blood cells, and as I mentioned before, or skin cells. So basically, research from a Japanese researcher called Shinya Yamanaka showed very nicely, and that's why the reason, by the way, that he got the Nobel Prize in 2012 for the research, showed very nicely that you can take skin samples from patients and or normal individuals, non-affected individuals, and you can trick these cells into thinking that they are stem cells. That's why it's called induced pluripotent stem cells, because you induce the cells in the skin to be stem again. You basically change the program of the cell. And now the cell not only thinks as a stem cell, but also behaves as a stem cell. And we call those, because we changed the program, we call them reprogrammed cells, because we changed the program. Now, the fate of this cell is going to be exactly the same as the stem cell. We only need 4 factors to do that. That was the beauty of the research that he published. So the advantage now is I can capture the patient's individual genome, the individual genetic material that the patient has within the cells. And I don't really need to go back to the brain and get the cells. I can have those stem cells now just by taking the skin sample. And these cells are going to have exactly the mutations or the genetic problems that the cell had to before when the brain cell is in the brain of the autistic kid. And the reason why this is important is because it doesn't matter. Remember I said we don't know what's wrong with autism, but we know it's genetics. So now we don't need to know. We want to know, but we can still have a living cell from a patient with autism that has exactly the same genetic problems that the cell had before. But now I can turn the clock back in time and look at the cells early on at the stem moment, before they make neurons. And why this is important? For instance, let's say we are walking around UCSD and you see a car stuck in the tree. You can ask, how did that happen? Imagine if you could go back in time and find out exactly how the car went there. Was it a tornado? Was it some bad joke from some kids that took a crane and put the car there? What happened? You can only find out what happened if you can go back in time for the early events before the car got stuck in there. And so you can have a better understanding of how the car got there. Moreover, once you find out why the car got stuck there, you can maybe even prevent it from happening. And that's exactly what we are proposing to do with the cells with autism, trying to model autism using stem cells. We are turning the clock back in time, so then we could be able to see the early events that the baby neurons have before they become fully mature, and what happened there that could be causing a problem for the brain to develop properly. So our model is to have cells from patients with autism, for instance, and also neurotypical persons that the individuals that do not have any neurological condition. And we turn the clock back in time and reprogram those cells, change the program of the skin cells into stem cells, and then we push them into neurons or differentiate them into brain cells or neurons and or other cells present in the brain. And then what we do, we compare them, how similar they are, how different they are. And if we see difference, can we do anything about it? So as an example, what we did, we used cells from patients with Rett syndrome, which is under the umbrella of Autism Spectrum Disorder. And we went ahead and took the skin samples, did the reprogramming, turned the clock back in time into stem cells, pushed them into differentiate into neurons, and looked at those neurons under the microscope. And what we see is neurotypical individuals, the ones that are not affected with Rett syndrome, had normal size body, we called cell soma. Rett syndrome individuals had small cell body. The other thing we noticed is when you look at the processes of those neurons, the brain cells, they have protrusions. Those protrusions are the points of contact, how the neurons communicate to each other. Those protrusions are also called spines, and they are where the communication between, the specific location where the communication between neurons is made. So we noticed that the Rett syndrome, autism spectrum disorder cells had less points of connections. Moreover, when we visualized those connections using specific dyes, we noticed that the points of communications were also decreased in Rett syndrome. Then we wondered what happened in a population of brain cells that have less points of communication compared to a population of cells that have more points of communications. And to do that, we used a specific type of dye that would go inside the neurons and will change the intensity or will blink when the cells are active. So whenever the neurons are communicating with each other, we will see differences in intensity. And the way we visualize this is by blinking. So if I could have the lights dimmed a little bit. Oh, thanks. So on the neurotypical or non-affected individuals on your right, you can see dots. And all the dots correspond to different neurons in the network. And everybody can see the blinking. The blinking corresponds to communication between neurons. When we do the same with cells that came from patients with autism spectrum disorders, the neurons are still there. You can see the dots, but the communication is not as efficient. Everybody can see there's less blinking. So we concluded then the consequence of having less points of communication in the network of neurons was less communication between neurons. So the communication is less efficient. And that could be underlying one of the issues early on during autism spectrum development. So we then asked the questions, how permanent were those changes at this early stage? So we added, for that, to answer that question, we added a molecule that is involved in cellular growth and development, and it's called insulin growth factor 1, short for IGF-1. And when we add this molecule in the system, we were able to rescue the communication between the neurons. So then they were more similar to the neurotypical non-affected cells. We were thrilled to see that. And we concluded that the defects that are present in brain cells at that early stage, in a dish at least, are not permanent. That is a very important point, meaning that early detection or early diagnosis might be really important. On top of that, it gives us insights on and better understanding on the disease, so we could start thinking about potential therapies. So in the near future, we plan to study other neurodevelopmental and other neuropsychiatric diseases in the lab, especially the ones that traditional therapeutic approaches are not available or are not well developed. And the reason why we think the therapeutic approaches are not well developed is because the diseases are not well understood. So to date in the lab, we have brain cells from patients with classical autism, schizophrenia, Williams syndrome, that's in a collaboration with our own Ursula, and also bipolar disorder, and more to come, because we think that we could discover very basic disease principles at early stages that could give us insights, not only how the disease works or how it starts, but also how to prevent and treat. So what I want you to take home as messages from my talk are, we used induced pluripotent stem cells, and they can be used as models, simplified representations of reality, to study autism spectrum disorder. We also can use this culture, this model, to gain insights into early stages of the disease, before the brain is already committed into being autistic, for instance. And lastly, we think that our research can be translated into drug discovery, because we can use such model to test pharmaceutical compounds. We did with IGF-1. Are there any other compounds that would improve the conditions of our cells? And lastly, I want to thank you all for coming. And specifically, I want to thank Rusty Gage for all the support and great mentorship. And I want to thank also these 4 girls that have helped me immensely during this project. And they are Ruth Offner and Anna Mendes, Diana Yu, and Yaneli Nunes. I also would like to thank all the Gage lab postdocs and staff members. They make everything run so smoothly and gave great, great contributions, both technical and intellectual. I would also like to thank Salk Stem Cell Core to keep us active and always learning a lot about stem cells and keep the stem cell culture alive in the lab. Leah Boyer and Travis Berggren. And I finally would like to thank our collaborators, Alison Watry from UCSD, Cassiano, his postdoc, Ursula Bellucci at Salk, Kristen Brannan, Tony Wishaboris, Eric Shen, and Dan Gashing. And thank you so much for your attention today.